for joining us tonight to learn more on EDI 200, the treatment for XLHED or XL Hypohidrotic Ectodermal Dysplasia. And leading our discussion tonight is Dr. Ken Huttner, and he is the Senior Director of Clinical Research with Edimer Pharmaceuticals. Dr. Huttner, I'll turn the evening over to you.
Thank you, Kelly, and thank you all for joining us. This is a great opportunity for me to update you on the work that we've been doing in our natural history studies and how that is going to lead us into the first clinical studies, which we are anticipating will take place in 2012. I've sent along a slide deck. It has 20 slides on it, so I'm hoping that people have access to that. I will read through some of it, but I won't read all the details, but they are on the slides, and they should be available through the website. So to start with, on slide 2, it's just basically a summary of XLHED, the X-linked form of hypohidrotic ectodermal dysplasia, which runs the whole range of issues from some that are serious and life-threatening related to overheating and pneumonias and many other issues which are very important clinically, but not in that life-threatening category, including those affecting the hair, the chronic dry skin and atopic dermatitis, the many dental issues, the dry eyes, and of course the psychosocial implications of all the clinical medical issues that these patients undergo. Now, what was important to us in looking at this disorder as we started heading towards therapy was to consider the variability. That clearly from the studies that were reported in 2004 by Rouse, based on several of the family conferences that the NFED held, there was significant variability in what appeared to be a series of boys and girls with HED. And part of what we've been doing for the last year and a half is trying to understand that as it would have implications for our clinical studies. So on slides 3 and 4 are really the foundation for everything we're doing. This is the mouse XLHED model, or the tabby mouse, and the dog XLHED model. And the data from them combined tells us so much about what is possible given the right dose at the right time, both in the antenatal setting, as shown in 2003 in the mouse, and in the antenatal and postnatal setting, as shown in both the studies in Switzerland in the mouse and at the University of Pennsylvania in the dog. When I look at these two studies, they're very exciting for us, and I know they are for you as well. But there are a few issues that we do have to keep in mind. We know that animal models can only take us so far, that with this unique disorder that has a relationship to development, that the development window, the period of development for all these tissues is very condensed in these animals. So, for example, a mouse goes from fertilized egg to weaning in 6 weeks. A dog might do that in 6 months, and humans do that, have that over many years. So that the effects that we see may take longer to achieve, and we may not be able to use the exact same dosing regimen in clinical trials. That's something we're just going to have to learn about. The second issue is that not all of the phenotypes that we see or the clinical issues that we see in our patients with XLHED are represented in these two animal models. For example, mice have only one set of teeth. So the issue of baby teeth and adult teeth can't be studied there. And finally, and this is turning out to be important for us, we know that in patients with XLHED, those who have the genotype that's proven to be an EDA mutation, there are many, many different mutations. At least 200 have been described. But in each animal model, there's only one mutation that's been studied. So the variability is much less within all the mice or all the dogs than we would see in the human population, and we have to recognize that and incorporate that into our plans. So we've done that starting with a series of human natural history studies that began in 2010, and they are started to summarize on slides 5 and on. We've done 5 studies now, 3 in the United States and 2 outside the United States. All total, we have data on 120 boys or men with XLHED documented by genotyping, and this covers almost 70 different mutations. So we really have a much better appreciation of the variability. And let me tell you a little bit about what we've done in these studies and how that's going to help us get into the clinic. So in 2010 in Colorado Springs for the NFED Family Conference, we started with our first study, and we were very fortunate to have Dr. Kathy Grange as our PI, and this was our chance to interact with the XLHED community and to get a chance to use non-invasive technologies to start to assess the very basic features of XLHED. Specifically, we were able to enroll 37 HED males and 25 control males, and we studied them with 2 new approaches. One was to actually count the number of sweat ducts, and this was done with a unique confocal microscope that had already been used by dermatologists for other reasons, other purposes, so that we knew it was safe. And at the same time, we could measure inducible sweating with a reliable sweat device that had been used for testing of cystic fibrosis. So both of these were taking safe devices to a new population. And you can see in the slide number 6 the distribution of ages. So we had many children and some adults with HED. And the first exciting results are shown on slides 7, 8, and 9. And here we see the images that were taken by the confocal microscope. So this microscope has an objective that actually sits on your skin surface, and those who've had it done will know exactly what I'm talking about. There's no pain involved, and it takes pictures below the surface of the skin with its laser light. And one can see on slide 7 what it looks like to see these beautifully outlined epidermal ridges, some of which, if they were on your finger, would be part of your fingerprint, but this is where all the sweat glands empty. And now, the rule of thumb has always been that under conditions of normal development, you have all the sweat glands you're ever going to have when you're born. And then as you grow, the skin spreads out so it looks like they're further apart. And that's what we see in slide 7. The 5-year-old on the left seems to have quite a density of these sweat ducts, which are the little white dots, and the 55-year-old on the right seems to have many fewer. These are actually the same magnification. So it just shows that even though these individuals have the same number per body, they have less per square surface area. So once we had that information from the controls, then we could look at individuals who didn't sweat, who had the genetic disorder, and we have 2 examples. One on slide 8 was done on the hand, and on the right is the picture of the father, who is unaffected, and on the left is a picture of his son, and this is all in the hand, which you can see the outlines of the epidermal ridges, and you don't see any of the little white contrast sweat ducts. So there are no sweat ducts and no sweat documented entirely with a non-invasive imaging. In the past, this would have been done by a skin biopsy. Not only can you do this on the hand, but you can also do it on the feet. And you can see in slide 9, on the right is the control foot with the epidermal ridges and the sweat ducts, and the left is an adult HED who has none. So we were able to show successfully that one could identify the sweat ducts and count them based on this area, and then we could combine that with the information on sweating. And the tool that we used for sweating was actually stimulating with pilocarpine so that the sweat ducts, however many you had, would be maximally active, and then measure the loss of water through the skin, or the humidity right above the skin. And again, those who were part of the study in 2010 will remember how this little device would sit on top of the skin for about 30 seconds. And the data is captured on slide 10. This represents all the data from the proven XLHED patients. There were 32, and the 25 controls, again, all males. And while it's a complicated slide, along the bottom is the number of sweat ducts in a certain area, and along the side or the y-axis is the net stimulated water release that could be measured. And if you look at the 25 blue squares, these represent the controls. They all have plenty of sweat ducts, and all but one responded to this pilocarpine stimulation with release of sweat. Now, none of them had any sweat issues. The one who didn't respond, we don't really know why he didn't, but he's totally healthy. At the same time, if you look at the 32 orange diamonds, you can see that 28 of them are in one cluster at 0 sweat ducts and 0 sweat. So that represents 85% of the individuals in the study, again, had no sweat ducts and no sweating. So that's the severe anhydrotic ectodermal dysplasia that people have described. However, there were 4 individuals in this study who had sufficient number of sweat ducts. They just didn't work in vivo, and in some of our testing, they didn't produce any sweat in 2 cases or reduce sweat in one of them. And one can see on the next slide, slide number 11, actually the real photos that are a little bit small of these 4 individuals. They're 4 boys aged 2 to 8, and you can see that they have plenty of sweat ducts as these little white dots. They may be patchy in their distribution, and they are variable in their function. So this supports the idea that there is a variability in XLHED, and that we may predict a different response to any future treatment depending on what the genetic mutation is. So as with other genetic disorders, we'll want to include everyone in our initial studies, and we may find that there is a connection between the specific mutation and the response to EDI-200 or any other replacement therapy. So this gives you a flavor for the type of data that we have been generating, at least in 2010. And I'd like to share with you 3 of the questions that we hope to address in 2011. We don't have all the final data in, but I think these are important and interesting pieces of information that are all headed towards the clinical study. So the 3 listed there, we're moving from skin to hair, as we know that that's an issue both in the thinness and the slow-growing hair. Also, we're trying to understand what are the clinical differences between HED and the non-HED ectodermal dysplasias. And finally, we have done an unusual study where we hope to look at how consistent the features of XLHED are between brothers. And the reason we want that is that when we move to the clinical study and we treat a newborn, we're going to be asked, how do we know if the treatment worked? Because there is variability. And in almost every case, that newborn will have had a prior brother with the same genetic mutation. So that if we can show that brothers are very similar, that then we hope to show that the treated brother will be very different in terms of his symptoms. So let me share with you some of the preliminary results. These are not published, but will be presented in this coming year on those 3 issues. And the first is on slide 13, and this comes from a study done with our PI, our principal investigator, Dr. Ophir Klein at UCSF, University of California, San Francisco. We did this in May, and the main part of this study was to look at hair, at how the hair structures were and how the hairs grew. And it's a nifty kind of approach, which is used in all hair studies, which is to take an area on the back of the scalp, to just trim the hairs very close to the scalp, and then 2 to 3 days later come back and see how they grew. And you can look at how many grow, how fast they grow, and how many hairs there are per little patch. And you can see in this particular slide that the Visit 1 and Visit 2 pictures are there for the same individuals, and there's 1 HED patient and 1 control. Now, this study was targeted at the teenagers and young adults, so age approximately 14 through 29, and we enrolled a little over a dozen of each HEDs and a dozen controls. And while I don't have all the data, I'd like to show you this fancy graph on slide 14, and I'll walk you through it a bit. This is the data from 24 of the subjects in the study. 12 are controls, and 12 have HED. And if you can see along the x-axis along the bottom, you see those 3-digit numbers. Starting at the left, the first 12 from 009 through 027 are our controls. And then from 001 through 023 are the numbers for the affecteds. And just starting with the controls, what you can see in this are these bars, and each different color represents the basic unit of hair in our scalp, and it's called a follicular unit. And the follicular units form before you're born, and they have a number of different hair follicles that grow in each one. On the average, for most of us, the number of hairs per follicular unit is 2. And you can see this in this graph where the bars represent percent of follicular units with 1 hair, which would be the purple bars, 2 hairs would be the green bars, 3 hairs are orange, and 4 hairs or more would be blue. And when you see that nice distribution of a few with 4, a few with 3, several with 2, and some with 1, that's a normal pattern. And you can see that in most of the control individuals. But when you look over at the HED individuals, and again, of them, they've all been documented to have actually XLHED in this group, you can see that they have the majority, if not all, of their follicular units have only 1 hair, and this really makes your hair look thin. When you look at the scalp, they have only 1 hair per follicular unit, so it's an abnormality of hair development. And I can tell you also that the hairs are thin and they grow slowly, which would not be a surprise to these boys because they tell us they don't have to get haircuts very often. Their hair feels a little fragile. And so this is a quantitative, non-invasive assessment of that. You may notice that for subject 008, that his pattern of the hair follicular units looks very much like one of the controls. He has many hair follicular units that have 2, some with 3. His average number of hairs per follicular unit is normal. What's interesting is that subject 008 has one of the same mutations that we saw in the summer study of 2010, telling us again that the phenotype may be very variable depending on your genetics. Moving to the next slide, which is number 15, we have some of the data that's coming from an amazing resource, which is the NFED, supported by Edimer, running this international registry for ectodermal dysplasias. It captures data from all subjects, all individuals who enter data on this. It's all ectodermal dysplasias, and as of May of this year, we had over 800 subjects who had entered data. And it's a wealth of data, and I share with you in this slide just some of the information that we can find. So this was data on males only, comparing XLHED, this is self-reported as XLHED, to the non-HED individuals, with fairly large numbers of both. And while their average age is very similar, you can see marked differences in this disorder. And they're highlighted there that the reports of families with infant or childhood deaths reflects what is in the literature, comparing XLHED to the non-HED. Genetic testing for both of them is less than 50%, which we hope to address. For those who have family members with XLHED, you'll know that this significant nasal congestion is very common in XLHED and relatively rare in the other ectodermal dysplasias. That XLHED is not commonly associated with limb or digit abnormalities, so there seems to be a very significant difference in the pathways that are affected, looking at 2% in the XLHED boys and 50% in the non-HED. It was striking to me to see that while XLHED males almost uniformly have minimal or absent sweating as part of the diagnosis, it's not uncommon for the non-HED patients to also have issues with sweating. That tells us that there is an overlap in the biology, that at some point we would need to see what the similarities are between the two and how we might consider treating some of the non-HEDs. It would be a different pathway, but there does appear to be some overlap in the biology. Quality of life issues are significant for both of them. So there's a great deal of data which is coming out. We'll be presenting this in the upcoming year as well, and we'll give you a summary at the summer conference in 2012. So the third issue relates to what we were looking for at the summer conference in 2011 in O'Fallon, and this was to look at the intrafamilial or brother-to-brother variation in HED. And again, Kathy Grange was a wonderful PI for us, and with the help of Kathy and the NFED staff and all those at Edimer, we had an amazing day of enrolling 64 subjects in one day. Now, for this study, we were focusing less on controls, but trying to bring in families. And it was the largest collection of families with HED that has ever been reported. We had 13 families with 2 affected brothers, and we had 7 families with 1 affected brother and an unaffected brother. All of these individuals filled out a detailed questionnaire. They had imaging of sweat ducts, and they had sweat collection. Now, the full results of what we find from this study will be presented at the upcoming conference in 2012, but I can tell you a little bit about these individuals. We started with 64, which was 52 affected. By the time we got through genotyping and full phenotype, we had 44 individuals with XLHED. Some of their issues, and it won't be a surprise, were hair thinning, which was reported by about 2/3 of the boys and the men, and most of them over 18. 92% of the individuals who are 18 or older reported hair thinning, so it clearly is a common issue. Dental problems were reported by nearly all. It's essentially 43 out of 44. It was very common to have dentures, and the great majority, on the order of 80% of those who did have dentures, had them by age 5 years. So this is telling us that the resources are getting out to the patients, and early intervention is clearly a part of care today. For those who are wondering, is it XLHED? Is it the X-linked form running in our family? In the families that had hypohidrotic ectodermal dysplasia, we had 12 families with 2 affected boys, and in 11 of them, so nearly all, we could find an EDA mutation, so they clearly were XLHED. We had 7 families with 1 affected son and 1 unaffected son, and in those, 6 out of 7 also had an EDA mutation. So in the populations that we've been studying, whether you have multiple affected boys or even 1 affected boy, it seems like the chances that it's XLHED, the form that we're studying and that we're hoping to treat, are very high. So that's great for us, and we hope that bodes well for our studies. We don't have all the data together on the concordance, on how similar the boys are, but I can tell you that in 7 of the families where they had the most severe mutations, that there was essentially complete concordance in the phenotype, so that you could have predicted what the younger son was going to have as problems from what the older son had. And so that would be what we need to share with the FDA to say, as we start our treatments, if that younger son now has treatment and develops sweating and has normal teeth, that's not coming from random change, that's coming from intervention. So again, we'll have all this information. We'll be presenting it at the summer conference, and I look forward to sharing it with you then. So that leads us to our slide 17, which talks about the next steps as we move into 2012. And first, just to reiterate, we have a huge debt of gratitude to all those who participated, without which we wouldn't have any of this data, we wouldn't have any of these studies. So thank you again if you are listening in. And I want to make sure that we address the issue of women with XLHED, because we've heard about it at every conference. We've promised that we will start some initiatives, and I can tell you that we have 1 going now and 1 coming soon. And briefly, the one that's going now is a survey that we're doing to get more information about the potential future for therapies. So we wanted to understand from women who were carriers, who were heterozygous for XLHED, how it affected them, how it affected their decision about having a family, and how they would feel if a treatment were available that were safe, that's number one, and that produced at least some benefit to boys or girls who were affected by XLHED. So that was a survey, a reproductive survey called XCores, and it's live. You can get information about it either from the NFED website under research or from the Edimer website under the patient area, and I'll give you a little update on it in the next couple slides. But that is up and running, and that's important for us identifying what issues we need to address and also getting contact information for individuals who want to hear about upcoming studies. In addition to that, we recognize that there are many women out there who believe that they are carriers based on having an affected son or an affected family member, but aren't 100% sure, and even if they are, may not know the exact mutation that they have. As part of our approach to that, we are going to be initiating a genotyping initiative, which will start with a pilot study that will offer genotyping to individuals. This will be women who are in their reproductive years and are considering that they may want to start a family or have additional offspring and would like to know something more about their risk based on their genetics. And this is an exciting time for us because these are, again, the potential mothers who will be having kids that we might treat. And we hope by the end of 2011 to have this started with at least a pilot where we will have women come to their PCP, to their physician that follows them, have blood drawn, and we will provide for the testing. All this will be done at a certified laboratory, and the results come back directly to the women. So those two studies are the ones that we've started in 2011, and you can hear most about them by joining the XLHED network, which also has a site to join on the Edimer website. And this is the best way to make sure that as information comes out, you will have it the moment it comes out. So for some of these studies, getting involved early is going to be important. Now, the next two steps really relate to what we want to do in 2012, and it's where we've been headed with EDI 200 for the last 2 years. And we're preparing to submit our data to the FDA that's required prior to initiating any clinical trials, which means treatment of any human beings with EDI 200. So the FDA has asked us to demonstrate in the usual fashion, that EDI 200 is safe. And at the same time, they want to know if we're going to be giving a new drug to neonates. They want to know how we chose the dose, when it needs to start, and how many doses to give over what period of time. So a lot of work has gone into this in the animal models, which are our best first guess. And then once we start bringing this into humans, then we'll modify that. And we need to recognize that this is an unusual program in that we have to do full safety profiles in both newborn animals, 2 species, and we have to provide information from adults as well. The FDA would like us to start with giving EDI 200 to a small number of adults, and with safety in the adults, then they would allow us to start giving it to neonates. This is the plan that we'll propose. 2012 should be the year that this happens. We will be sending all the safety information and the plan to the FDA, and then the FDA helps guide us as to the approach they think is most appropriate. So we anticipate that this will be coming up, and certainly we'll have an update at the summer conference. So finally, on slides 18 and 19, I do have a little bit of summary of this XCores survey. I wanted to give those who are interested in the woman's side, the carrier side, a little bit more granularity as to what we were doing. This describes a survey that's being done on reproductive history and attitude. It's being run through a genetic counselor that's working with us and our Edimer team. It was launched shortly after the summer conference this past summer, and in less than 3 months, we have nearly 200 respondents, women who've completed the survey. And on slide 19, I give you some of the results, particularly for the women of childbearing age. We have 110 that have responded. The survey is in English only at this point, so it's no surprise that nearly 80% are from the U.S. But it does reinforce that many of these women have had affected sons. A few of them have had genetic testing, which we'll address in the genotyping initiative, and that many of them have had decisions about their reproduction and future family size that are negatively affected by their concern over the risk for XLHED. And as we talk to more women and hear more about it, it's reinforcing the idea that if we have a safe therapy that corrects at least some of the symptoms of XLHED, the great majority of women would consider both prenatal and postnatal dosing, and the numbers don't differ much as to whether it would be an affected boy or an affected girl. So this reinforces that there's a interest for EDI 200 as an intervention which may correct, and depending on how close the humans are to the animal models, it may correct many of the features of XLHED and bringing it forth in a safe but prompt fashion. So we look forward to more data. Again, this will be presented this coming year, and we'll certainly discuss it during the summer as well. So please, if you are in this age group and you are interested in adding to the information and helping us guide through where we should go and what should be our series of steps, that would be great. So that really brings to a close a summary of what we're doing, of where we are, what's planned for 2012, and I've put a picture of the Edimer crew on the last slide, number 20, hoping to see as many of you as possible at the Family Conference in 2012 and to share a full summary of what we've done in 2011, and hopefully we'll be in the midst of moving into our clinical studies in 2012. So thank you for listening, and I hope we're near to being on time, and I'm happy to take any questions.
Thank you, Dr. Huttner. Always very enlightening to hear you speak and hear the updates on EDI 200. I know it's exciting information not only for the families that are affected by XLHED, but those that are affected by other of the syndromes as well, as the progress of research just keeps moving forward. Very exciting times. So I'm going to unmute everyone's lines, and if you have questions, this is go ahead and speak up, and feel free to ask Dr. Huttner any questions. Mary Fate from the NFED is also on the call if you have questions for her related to the treatment, and myself as well.
The conference has been unmuted.
Okay, does anyone have a question?
Yes, this is Danella Anderson, and our son participated in the survey this summer, and I was wondering when we would be getting those results from those tests this summer.
Sure. So the genotyping tests have all been done. So if your son was one who had blood sent, that information is being sent to Dr. Grange, and she should be communicating with you in the near term if she hasn't done so already.
Okay.
And, I talked to Kathy Grange, I guess it's been 2 days ago. She does have the results. She's got a lot of phone calls to make, so she's in process. So just be a little patient, and she'll be in contact with you real soon.
That's awesome. Thank you very much.
Sure, our pleasure.
Any other questions for Dr. Huttner?
I have a question. This is Laura, Caleb's mom. Where can we refer to, and I may have missed it in the beginning, where could we refer back to the slides that you're referring to?
These slides, if you still have the invitation or the reminders from for tonight's meeting, there is a PDF which is on the right column of that email.
Okay.
And you can download that to your computer.
Oh, great. Thank you.
And if we don't still have the invitation?
You can send me an email. Send an email to Kelly, and I will resend it to you.
Okay, great.
This is Danella Anderson again, and I don't want to butt in if someone else has a question. We have a daughter that participate in the survey, and I am a woman, of course, above childbearing age at this point in time, but I think that there's a possibility I may be a carrier. At some point, will you be doing any kind of testing for individuals past childbearing age?
We don't have that as part of our initial study. I think we need to do our pilot first to see what kind of results we get. And at that point, we'll have to address that. But I will put that on my list. One of the things that will happen is that as we do more of these tests, the price will come down. So, we'll be able to do more for people who want to find out. And the other issue is that if you already know the mutation that might be in your family, it's much easier and faster and less expensive to confirm in another family member.
Okay. Thank you.
Sure.
I apologize. I do hear some feedback as we are speaking, and I don't know if that's because all the lines are unmuted. We haven't had that problem in the past. Sometimes it's one person's line that could cause that. And unfortunately, there's just not a way to figure that out. Are there any other questions this evening?
I have one. This is Janice from Oklahoma, Jill's mom. Did I hear you right in saying that y'all were going to start on adults first with the protein, and then once you prove that that's safe, then you'll go to the newborns and such?
Exactly right. That's what we discussed, and the FDA, they recognized that we're not expecting to see any benefit in adults. They just felt uncomfortable that this was the only drug out there that anybody's working on that would go right to a newborn without doing even a short, small adult study. So you're exactly right. We're going to get some data on safety. We'll also know something about what levels are achieved in the blood. So we know from the dog model what kind of levels you need to get a response in dogs. And we want to convert that to what we get in humans, and we'll learn some of that from the adults. But we want to move through that part of it quickly, get all the safety data that we can, because we know that things are very different when you treat newborns, and the only way to find out once you're comfortable with the safety data is to get in there and treat newborns.
When you inject, this is Marcy Barbet in New York. When you do the trial on adults first, what sort of safety issues? How long would it take for you to say, okay, nothing's wrong, or oops, there's a problem?
Right.
Do you have to wait a certain amount of time?
Yes, it's a great question. And what the usual standard is for these very first or phase 1 studies is to try to look at whatever might happen when your body is at the highest level of the medication. So for an intravenous administration, it's right at the end of the administration, you're at your peak level. Mm-hmm. So after we've given all the doses, we would do whatever blood tests that are the usual tests that the FDA wants to see right at the peak exposure, and that would be the first point at which they look for safety endpoints. So assuming these adults are feeling fine and there's no sign of a problem right at the site where the IV is and all their blood tests are normal, that's really the acute dosing that they're looking for. Mm-hmm. We would follow these adults out longer till it's out of their system, so that would be probably another week or two, and then another month or two after that for other issues. But the real acute safety, if there's going to be a sign, it's usually going to show up when you're at your peak exposure.
Right. Okay. And the protein is a natural substance. It's not a man-made.
Well, no, it is a man-made. It's the combination of a human antibody at one end and EDA-A1, the molecule which stimulates the whole pathway at the other end. So it's not something that's found in exactly that form in nature. And in addition, to make it work, they have to come in clumps, because that's what is supposed to happen inside the body. So we've made it that way so that we know it works, but it is something that the body wasn't used to seeing. We know that when you give that intravenously to dogs, they're happy with it. It doesn't cause them any problems. This is in the XLHED dogs, and they get a response and they do well with it. And that's with single dose and with multiple doses, and that's what Maggie has published, and that's really where we're going to start when it comes to the dosing.
Okay, great, thank you.
You're welcome.
We have time for a few more questions, if there are any tonight.
I don't know if this question is too technical or to get into, but I am interested in what is the part of it that's an antibody? Why do you have that connected with the EDI protein?
Right. So it turns out that it's important that these multiple molecules of EDI-200 wrap around each other so that they can be stable and they can do their signaling to tell the skin to develop the sweat ducts and the teeth to develop. And when you to get it to wrap, you have to attach. They have to attach to each other. And so the piece that was added had two functions. One this is from the human antibody. One is so that there can be these bonds between individual EDI-200 molecules in a stable fashion that can circulate through the blood, and when they get to the right place, it'll still be this combination of somewhere between 6 and 12 copies coming in together.
Okay.
So that was one of the real good reasons to do it, because if they fall apart and they go their separate ways, each of the molecules, then you lose all activity. So the normal molecule does that with some regions like collagen in the molecule, but that doesn't work well when you try to make it in tissue culture. And the other reason, which I thought was a very smart design, I didn't have part of it, that was done by our group that we work with in Switzerland, was that the piece that was attached binds specifically to receptors in the human placenta. So they were thinking ahead that someday they wanted to have this molecule built so that it would transfer across the placenta, thinking of antenatal treatment. So we can't get there in a hurry because there's so much safety data that we need to generate first. But having that, we're hoping will allow it to be, to go from injection into a pregnant mom to her affected fetus.
Okay, so that's to get from the mother's blood. So you put it into the mother's blood, and then it gets into the placenta so the child can have it.
And it goes from the placenta then into the child's blood. So those are the two purposes.
Okay.
It stabilizes everything, and it also has the potential to work in, and that's when they injected it in the mice, they injected it into the tail vein of the pregnant mice, and it went to the baby mice before they were born, and when they came out, they were, except for some hair, they were totally corrected.
Which is also probably better for the safety of the children because you don't have to inject it directly. That would be a lot more difficult.
There are pluses and minuses to both of them. We certainly, when we do this study in neonates, we will be using, a center where they know exactly how to deliver this, and that, as challenging as it is, and I'm a neonatologist, so I'm used to putting IVs in and injecting into small babies. It will only be people who are experts in doing it.
Okay. Thank you.
Sure.
Doctor, it's Marcy Barbet again. You said a second ago that there were still hair issues with them. Did the injection not cure or solve the hair problem for either the mice or the dog, or just partially?
Well, if you inject after they're born with the current protocol, the dogs did not improve their hair, and the mice had a little bit of improvement in the hair on their. No, they had no improvement in the hair at all. But remember, they have a very different hair system than we do. They have multiple hairs, they have guard hairs, and all this kind of stuff. But it did appear that having an effect on the number of hairs seems to require an exposure earlier in development than we've seen with the dog.
Mm-hmm.
But in the mice, when you inject them before they're born, it fixes the hair.
Oh, okay. Okay, great.
Any other questions?
Well, certainly if anyone comes up with additional questions and you can send them to Kelly, they'll all make it to me, and I'll be happy to respond if you think of something after the call.
Thank you. Thank you. What great information, great questions are being asked tonight. Anyone think of anything?
I got one more question.
Sure.
When do you think, if everything, works out and is successful with the trial and, goes, but successful, I guess I mean that, you see that this is actually, making a difference in the health, the children, when do you think, how long would it take before it would be available, for instance, to see if it would help children who are already born but, obviously are still developing?
Right, and this is, I was, throwing that out or suggesting that we need to take that into consideration when we think about how long development takes in a human, where a dog has a full set of adult teeth when it's 6 months old, and yet our kids, they're 10 years old and they're still developing teeth. So we do feel that we don't know everything about developmental biology, but there might be the opportunity to have some impact. And we are going to be doing more animal studies to ask that question, but it really won't have a clear answer, I think, until we get some data from the newborns that are treated and we follow them throughout the first months to years of life. So my hope is that after we've worked into the first two studies, the adult safety study and the neonate safety study, that we'll be thinking about additional studies and what we might be able to do. So, for example, we looked at teenage boys and boys in their 20s to see what goes on with their hair. And we know that their hair is not, the hairs that they have are not behaving normally. So that's something that you could potentially have an impact on even at a young age. So we'll be thinking about those. We're going to get the first two studies up and rolling, and then that's something that, with the right resources, that I think those are questions that you're great to point out, because those are things that I think deserve answers.
Yeah, and I'm thinking, even more particularly, with, for instance, some of the respiratory issues, like nasal congestion, or I think on one of the pictures it had something about the glands in the esophagus.
Right.
And so, what is the effect of that for small children
and
their development?
Because there's so many great questions. So many great questions to answer, and we just hope that we'll continue to have the success and the resources to address them all and address them as quickly as possible.
Okay.
If your trials are successful both in the adults and the babies, how long does it usually take before it gets to market so that new pregnancies can be affected? Is it years or is it months? I mean, if everything works perfectly, when can we see this actually starting to get out to the general public?
Right. So you can understand one of the challenges is that this is not going to be a placebo-controlled study.
Right.
So it's not we're going to have half a dozen untreated and a half a dozen treated to compare to each other.
Mm-hmm.
So we're going to be looking closely at what you can follow and how striking and consistent the benefit is in those who are treated. Mm-hmm. In the beginning, in the first study of neonates, we are going to start with a dose which we know works in dogs, but we have so much to learn about what is the right dose to use in humans that it's going to be challenging to know, to hit it spot on, from the very beginning, and to have the kind of results, even without a placebo group, that are totally convincing, both that they come from the drug and that they are a meaningful clinical benefit. But those are the two things that we need to prove, and that's why we do so many of these natural history studies, so we know what to expect in the first months or even years of life, and that if we can do better, that we can be convincing in showing that it's coming from the drug, that it's long-lasting, and that it's a real clinical benefit. When we can do all those things, then that's the point I think that the FDA really wants to look at and wants to bring this to market.
Right.
But that's a lot for us to do, and sometimes you're lucky with the very first dose, that you're using the right dose and the right time, but more often you need to test some different doses, a higher dose, a lower dose, more doses, to get to the kind of result that they want to see. So that's not a definite answer, but those are the things that we need to show and that the FDA will want to see in order to make it widely available.
Thank you so much. Dr. Huttner, can you remind us of two things? One, you're going to be at the 2012 National Family Conference, correct?
Exactly.
And number two, how do our families access the XLHED network?
Right, so if they go to the Edimer website, if you search for Edimer or you Google edimerpharma.com and you come onto our main page, I think on the right-hand side there's a link that says XLHED Network. And if you click on that, that gets you to where you can sign in on the network. And if you put in your contact information, that means any study, any information that goes out, you will be getting it as soon as anybody else. You'll know right away.
Thank you. And you'll also want to make sure that your contact information is updated at the NFED office, especially your email address, because when Edimer Pharmaceuticals contacts Mary and says, this is what we're working on, and we're looking for people between this age and that age, or whatever the parameters are, she will be contacting you through the means of phone and email. So you want to make sure that NFED has that contact information. We thank you very much for your time tonight and sharing all this wonderful information with us. Thank you to all of you who joined us.